I will be introducing you to the new Ensembl Genome browser. And before we do that, I'd just like to give you a quick introduction of EMBL and EBI. So EMBL stands for European Molecular Biology Laboratory, and we have 6 sites across Europe. So EMBL-EBI is where we're based, and this is in Hingston near Cambridge in the UK. Then we have a site in Grenoble, France. These people mostly focus on structural biology. We have a site in Barcelona, and they are working on tissue biology and disease modeling. And then there are 2 sites in Germany. So we've got Hamburg, people working on structural biology again, and then Heidelberg is all of life sciences, and this is the headquarters of EMBL. And then we have our site in Italy, in Rome, and people there are working on epigenetics and neurobiology. So EBI is part of EMBL. So EBI stands for European Bioinformatics Institute. We are the second largest EMBL site. So we have over 800 employees. We provide free bioinformatics resources. The research groups are mostly focusing on computational biology and bioinformatics training. So EMBL-EBI offers a number of resources that you guys might already be familiar of. We have Ensembl, which is one of the most popular and the most used EBI resource, and we will be talking about Ensembl today. But you might have also come across Open Targets or UniProt. These are the protein people. EuroPMC, which provides open access literature, and then we also have European Variation Archive, AlphaFold, ENA, and so on. So the learning outcomes for today are that by the end of this webinar, participants should be able to access the main types of data through the Ensembl website. This could include genomes, genes, and also I'll show you where you can find help and documentation on the new website, as there is lots of it available. If you have any questions at any point, please do use the Q&A tool, and then my colleagues George and Luis can answer them live. So the first sequence was sequenced by Fred Sanger and his team in 1977. So this was a small bacteriophage genome of about just 5,000 bases. And this is what really kickstarted the era of sequencing and sequencing more complex organisms. So since 1977, where the bacteriophage was sequenced of about just 5,000 bases, this then paved the way of the draft of the human genome, which was published in early 2000, so in 2004, which was a much larger genome of about 3 billion bases. And since then, many other organisms have been sequenced, and today we can sequence the entire human genome in a matter of days. So sequencing creates these, or produces these, this data. So this is, if you look at this slide, it's just a string of letters of genetic code. And if I just look at this data, it doesn't really make any sense. So for this data to make sense, we need to process this data and we need to work with it. And this is what genome browsers do. So Ensembl is a type of genome browser, and we take publicly available sequencing data and we make sense of that data. So we annotate it with features like genes and variants. We perform our own analysis on this data, such as comparative genomics analysis. So Ensembl annotates and maps genetic features from genome sequences, and we also bring together information from various other databases to make it available in a single site. We have many different features that are annotated by Ensembl. These include genes, variants. We have regulatory features like promoters and enhancers, binding sites, and many more. As I mentioned, we have variation data for a number of species. We offer pre-computed comparative genomics data as well, such as homologs, gene trees, whole genome alignments. And then we also allow you to export our data. So you can do it in sort of like a more customizable way by using Biomart. To use Biomart, you don't need any programming knowledge. You can just use it on the website. Then we have other tools for your data processing, such as Ensembl Variant Effect Predictor, VEP. You can also display your own data in the browser, and we also give you programmatic access to our data via our APIs. Most importantly, Ensembl's data is completely free, so it's open source, and you have access to our whole genome flat files via our FTP site and also our source code via GitHub. So we've got a few human genome assemblies that are available in Ensembl. So we've got the new T2T assembly available in rapid release. We've got GRCH38, which is the current human genome assembly that is available on the main site. So it's available on www.ensembl.org. We also support the previous human genome assembly, so GRCH37, and you can find it on grch37.ensembl.org. And then we also host the even older human genome assembly, which is NCBI 36 or HG18. It does have quite a few gaps and it is no longer updated, but you can find it on ncbi36.ensembl.org. Then we have non-vertebrate species available in Ensembl genomes. So we have Ensembl plants, metazoa, bacteria, protists, and fungi. We also have a site dedicated to COVID-19. It doesn't have all of the functionality that the main website has, but you can find the annotation for SARS-CoV over there. I did briefly mention rapid release. So rapid release is sort of like a lighter version of Ensembl, and this site was made available to keep up to date with the constant release of the new genome assemblies. So we call it Ensembl Rapid Release. It's updated every 2 weeks. And this browser has limited information. It includes genomes with gene annotations only. It does have the BLAST tool available, and there are some comparative genomics data that is released. However, it has now been frozen because all of the data that we would have added to rapid release will now get added to the Ensembl beta site, so to the new Ensembl site. And we will move on to that in just a few seconds. So we are continuously aiming to improve the Ensembl resource and the user experience to support research in comparative genomics, in evolution, sequence variation, and transcription regulation. So since the launch of the Ensembl website, there have been many more organisms that have been added, and our understanding in genomics has expanded since then. So the amount of genomics data constantly increases. And with that, the Ensembl database keeps increasing as well. So as you can imagine or know that if you're an Ensembl user already, that there is massive amount of data that has great impact on the performance of the browser. So on top of that, if you need to navigate the different Ensembl site, depending on which genome assembly you're working with or you're interested in, it does sometimes makes comparison a little bit more difficult. But please know that we are aware of these performance issues and we are working on a solution on this, and we're working on making the database future-proof. So in the past few years, our Ensembl developers have been working tirelessly on the new Ensembl site. We're calling it Ensembl Beta at the moment. And it has a more modern look and better performance. So this is what the homepage looks like, and we will do a demo in a few minutes. So the Ensembl Beta site has gene annotation and analysis that is provided at increasing rates. It is an improved website for an improved user experience. and we provide a comprehensive suite of methods to access Ensembl hosted annotations in standard formats. The infrastructure continues to provide the community with access to more intuitive interfaces and new future developments. It is highly responsive. As I mentioned, it also has a modern user interface. The users are going to have the ability to browse multiple species of interest simultaneously. And the Ensembl Beta is going to bring together Ensembl Rapid Release and Ensembl Genomes. So these are all non-vertebrates and vertebrates in just one single site. It is under testing, so it's under beta testing. So we would like to hear your feedback and thoughts. So we're aiming to release the new Ensembl browser in 3 development phases. So the first is the beta version of the website that was delivered in 2022, and this provides genome browsing. It allows viewing attributes of key entities and also search, blast, and help pages. Then the second stage is minimal viable product, so MVP, which will provide equivalent functionality to the Ensembl Rapid Release site, including variation visualizations, homologies, and support for all new genomes. The Ensembl Rapid Release site is then scheduled for retirement. The third stage is a new website. So the new website functionality will reach that of the current site. So the main, the Ensembl site that is currently available. The ensembl.org or the current Ensembl websites are then going to receive minimal updates and will be archived in due course. I mentioned that the new Ensembl browser has an app-based approach to functionality, so you can explore assembly and sequence information using the species selector. So on the new site, you'll be able to select multiple species at once and explore them across all apps. The Genome browser lets you visualize the genomic region, map annotations via tracks, and explore genomic features in the genomic context. If you want to learn more about a specific feature, for example, if there is a transcript that you're interested in, if it is a BRCA2 transcript, for example, then you can find more information about that transcript, about that gene, by using the entity viewer. In the entity viewer, you can also explore gene relationships, for example, homologs in other species. Then we also have tools, so you can already perform BLAST queries on Ensembl Beta. The power of the BLAST tool via the new site is that you can now BLAST across divisions. So for example, you can now align a bacterial sequence against a fungal sequence. And then we also have Ensembl Variant Effect Predictor available, VEP, which allows you to annotate your own variants and identify which genes, transcripts your variants have affected. If you need help on the new site, we have several options. So you can open the general help and documentation pages, which include guides on how the data was generated. You can also access data and tool-specific help pages, which are available throughout the different Ensembl pages. So whenever you see sort of like a small question mark icon like this, you can click on it and you can find the tool-specific help pages. You also have the ability to send us an email via the contact form, and this will put you in touch with the outreach team. We also have 99 human genome assemblies available and their annotations on the beta site. So these include the older GRCH37 assembly and also the current most supported GRCH38. We also have the most recent T2T assembly, and then you can also find the genomes of 48 individuals or 96 haploid assemblies from HPRC, so from Human Pangenome Reference Consortium. As the name suggests, the new Ensembl browser is under beta testing, so it is still in development. Our colleagues are working hard on making the new features and data available on the new site. Some features you can expect to come across in the future are more gene expression, ontology, and pathways data, phenotypes, additional variation data. We already have the web available, and then you can also expect regulatory data and also XREFs, so external references. And with this, I'm now going to move on to the demo. So you can find the Ensembl website or the beta website on beta.ensembl.org. And this is what the homepage looks like. So at the top, we have the different apps available. So you have the option for search. So this is site search. Then we have species selector. We have genome browser, entity viewer, blast, web, and help and documentation. So to start with, you can click on species selector, either from here on the main section, or you can click on species selector at the navigation bar at the top. Once you're on species selector, you can then enter the name of your species and search for it. We do have some popular species shown here at the bottom. So we have the 99 human genome assemblies over here, and then we've also got the mouse species, and we've also got zebrafish. As I mentioned, plants, metazoa species, protists, fungi species, bacterial are all going to now be available in just one single site. So that's why you can also see bread wheat. So you can see the wheat genome and also the Japanese rice, tomato here, and so on. Let's say that I'm going to search for a human. I've entered my species name, and then I'm going to click on find, and I get my results over here. So I've got all of the human genome assemblies available. To select a species, you can just click on this box and then click on add. You can select multiple species at a time, as I mentioned. So you can also, for example, find mouse, and then I'm going to select mouse reference. So the C57BL6J, this is GRC M39. Once I've selected that, I can just click add, and I've selected human and mouse as it's shown at the top over here. To find out more information about a genome assembly, you can click on its name. So I've clicked on human, and now this is going to take me to the human species page where it's showing me the number of assemblies that are available, so 99, and then within the GRCH38, the number of coding genes, the number of non-coding genes, pseudogenes, the homology information variation, so a number of short variants that are present, and also some regulation information as well. You can also now, if I would like to go to Genome Browser, I'm going to click on this app button at the top. And then I'm going to search for a gene name. I'm going to choose BRCA2 as an example. Enter the name and then click on Go. So I've got results over here for for mouse. But if I want human results, then I can click on human, and then I can type BRCA2 again. And if I click on Go, I've got my BRCA2 human gene. To find out more information about it or view it in the Genome browser, I'm going to click on this gene. And again, I get the option of whether I want to view it in the Genome browser or in the entity viewer. I'll start with Genome browser by clicking on the Genome browser button. And this is now showing me the BRCA2 gene within the genomic context. So I can also see the neighboring genes. I've got BRCA2 just over here, and I can click on +14 to view all 14 transcripts of the BRCA2 gene. Everything blue that you see on the side is clickable. So all of these transcripts that you see are interactive. If I click on the transcript, the first one, for example, it's going to give me the transcript ID. It is a main select transcript. So main stands for matched annotation between NCBI and EMBL-EBI. We have the biotype. It's on the forward strand, and then we also have the coordinates here. Then we have some further information about the BRCA2 gene, and then you also have the option for downloads. So if you click on downloads in this pop-up menu, you can then see that we'll have the option to download the genomic sequence of the transcript, the protein sequence, the cDNA, CDS, and exon sequences. Or you can download the gene sequence, so the genomic sequence and the exons. To jump out of this menu, you can click on anywhere outside the menu. Then on the right-hand side, you can click on this little arrow over here, and then that's going to show you right-hand side information section. So we've got BRCA2 over here, the number of transcripts, and then we have information on, you can say that you only want to view the protein coding genes or protein coding genes on the reverse strand. So you can sort of like manipulate your view from here. Then we've got some assembly information tracks over here available as well that you can choose from. We've got variation information at the top on the right-hand side and also regulation information. If I come back to my transcripts and I'm going to click the first one again, you'll notice that in the menu, you again have the option to view it in the Genome browser. So you can zoom in further on this, or you can view it in the entity viewer. So let's do that. Let's go to entity viewer now for this transcript. This is the main transcript for the BRCA2 gene. So now I'm on the entity viewer. And from here, I've got my transcript information. So we have options for more information. So we've got information on RefSeq match, the CCDS match, the protein ID that this transcript matches to. Then we also have information on the combined exon length and the number, the coding exon that this is on. We've got some download options as well. So you do have the option to download the genomic sequence for this transcript, the cDNA, CDS, exons, and protein sequence. And then we also have options or buttons at the top to view gene function. And this page is going to give you further information on the proteins, the protein matches or the protein information for this transcript that comes from Panther and PFAM, and we've got some UniProt and SwissProt information as well. With time in the future, we're also hoping to add variants information over here, phenotypes, gene expression, gene ontology, and gene pathways. I'm going to go back to these buttons, the blue buttons at the top, and I'm going to click Gene Relationships. And this is some comparative genomics information we've got, so you can view the BRCA2 gene in other species. So we've got, for example, the common names. So we've got guinea pig, blue whale here, their scientific name. We've got information on protein similarity, percentage coverage, the gene symbol, and the corresponding gene IDs in the different species. I'm going to move back up to the navigation bar at the top. So we've explored the species selector, we've explored Genome Browser, and we've also viewed the entity viewer. I'll quickly show you BLAST, as the BLAST functionality is available. So you can select multiple species at a time. So you can do that by clicking on Select Species, and you can enter your sequence over here. You can choose between nucleotide or protein sequence. The database that you can choose from includes genomic sequence, transcripts, and proteins. And then we also have the web tool available. So this is Variant Effect Predictor. And from here, so you can use the web tool to annotate your own variants. So for example, if you have done your sequencing analysis, you've called your variants from that analysis, and now you would like to find out more information about your variants, you can then upload them into Variant Effect Predictor. And then web is going to give you information on where these variants fall, what genes and transcripts your variants have affected. If they're already available in the database, then we will give you information on their allele frequency, so population genetics data, phenotypes associated, any pathogenicity scores, and so on. And then finally, you also have the option for help and documentation. So from here, you can find help on how to use the species selector, the genome browser, the entity viewer, and then we'll also keep adding to these pages on the different tools as they are made available on the site. I'm just going to move on to the chat and the Q&A section to see if there is anything that I can demo on the screen. So there is a lot of options available to download the data that we're showing on the Ensembl sites. As I mentioned before, you can go to the Ensembl the Genome Browser app over here, and then depending on the type of data that you would like to download, you have various different options. So you can either click on the transcript and then click on downloads, and then you can download the different types of sequences available. Or you can also go to the entity viewer, which is just over here. And then you also have, depending on the page that you're on, you do have the option to download the data from those pages, either by clicking on them, or you can click on this downloads button over here. So it's sort of like this, a downwards pointing arrow, and then select whether you want to download the genomic sequence for this gene, the exon sequence, or for the transcripts, or in case of transcripts, you can download the cDNA, CDS, exons, and protein sequence. So there's a question about how to locate genomic sequence with protein sequence. So if you are looking for a specific transcript. So you've searched for your gene and then you're interested in a specific transcript. So we give you protein information matched to transcripts because proteins map to the genome assembly via the transcript object. And that's why you can find some protein information by clicking on this transcript tab. So if you click on the transcript tab, this is where you will be able to find some protein information as well. So on the transcript tab, we've got, for example, this main select transcript selected. And then if I scroll down a bit where you see download, you do have the option to download the protein sequence that is associated with this particular transcript. And if I click on gene function, this is also another way to find some protein information. So after clicking on gene function, you will then get this option. It's not currently available, but you do get these different options, so different pages that you can scroll through. We've got proteins available over here, but then you'll also get variants, phenotypes, gene expression, gene ontology, and gene pathways. The question was about proteins. So for protein information, we've got information from Panther, PFAM. We'll link you out to UniProt, Interpro, and also PDBE. And then from here, again, you do have the option to download either the protein sequence or the CDS sequence. So you can click on one or the other and then download that. So there's also a question about if there is an option to visualize associated genes like nuclear mitochondrial dysfunction. We will make available more data on gene pathways. So you can see sort of like the different pathways that the gene is involved in. We'll also make available gene ontology. So this is molecular function, biological processes, and also the I've forgotten the word for it, but the gene ontology information that tells you exactly where in the cell that gene is located. So we will give you all of that information eventually in this gene ontology page. And then in gene expression pages, you'll also be able to find out which tissues or which experiments have found out different tissues or different organs that these genes are highly expressed in. So you can sort of like use all of this information to then sort of like visualize the gene in different aspects. Yes, so there's a question if you can see data variation by using the dbSNP information like RS codes. Absolutely. This is currently available in the main site, so you can definitely search for RSIDs, and this will also be available in the beta site as well. So you can definitely search for RSIDs. But apart from variation data, you can also search for phenotypes in the main Ensembl site, and I'm sure this functionality will also be available in the beta site, that you can search for phenotypes, and then we will give you information on the different genes that come up or variants that come up that are associated with that specific phenotype. Is it possible to get bar plot of one gene expressing in different organs or different species? If yes, how? Yes. So this will become available. This is going to become available in this gene expression page. And this is where we're going to show you. Currently, on the main website, you can find gene expression data in sort of like a heat map where we have our experiments that we have the data for and the tissues that these experiments were performed in. And then we show you if the gene is highly expressed or low expressed in specific tissues and organs. There's a question about functional predictions. So is it possible to get protein functional predictions from PFAM, Panther for other transcripts other than main select? I believe so, yes, as long as there is information from PFAM and Panther, then we do associate that with the transcript. So if that data is available, then we do try to associate that data with the transcripts in Ensembl as well. And then we will give we can also link you out to, for example, Interpro or PDBE for that protein functional prediction. Craig's asked, what is the anticipated time frame for getting the full new browser with full functionality? So we are at the MVP stage right now. So where all of the rapid release data should now be in the beta site. and we are working now towards the third stage. Let me just go back a bit. So the third stage, which is where the new website functionality is going to match the current Ensembl site. This is going to take time, but we're hoping, well, we've set an internal deadline until autumn. So someone's asked if they have a CDS sequence, can they identify the protein? Yes, you can use BLAST for that. So the BLAST tool is available just here at the top. And in here, you can enter your sequence. So this could be plain text or FASTA. You can select your species that the sequence comes from, and then you can use the BLAST tool for sequence similarity search. And yes, definitely, as George has mentioned, for regular updates, please be sure to follow our different pages, our blog, which is on ensemble.info. But what I will do now is please do keep the questions coming. But I can just move on to some of the closing remarks, and then I'll leave some space for Q&A. So we've seen the Ensembl, the new Ensembl website. It is still under beta testing, and this is why we would love to hear your thoughts and feedback. So please do, if you have time later on, go and play around with this new site and let us know what you think. Let us know if you're having issues somewhere, things that you like or things that you don't like. You can do that by clicking on this feedback button. So it's sort of like on the left-hand side at the bottom. I've highlighted it in this orange square. So you can use this to rate your experience and give us your thoughts. And if you have enjoyed the webinar, then please do recommend us to your colleagues. We'd also do a full browser course. So this is a one-day course on the Ensembl browser, and this is aimed at wet lab scientists. Apart from that, we also do a REST API course. So this is sort of like a half-day course on Ensembl REST API, which is aimed at bioinformatics, and it shows you how you can access Ensembl's data by using different endpoints. And REST API is language agnostic, so you can use Python, R, Java, whatever language you're comfortable with. We also do Train the Trainer course, which is a one-day course on how you can deliver Ensembl training. We can teach an Ensembl course at any institute. The courses are free. However, for high-income countries, we do ask for trainers' expenses. So if you are interested in hosting us, do get in touch with us on helpdesk@ensemble.org. Or you can find more information about how to host on training.ensemble.org/hosting. George has mentioned this in the chat already, but if you'd like to keep in touch or keep up to date, do follow us on Facebook. We're quite active on LinkedIn and Blue Sky, so you can find us there. And then for detailed information on updates, new release, you can check out our blog on ensemble.info. We also post about jobs and internships on there. And we also have these monthly posts on the team where you can meet the different people from the Ensembl teams and sort of like get to know them a little bit better and see their career path. If you do use Ensembl, please don't forget to cite us, as it helps us understand who uses our resources and how they're using it. So we've got our latest publications on the slide over here, but for a full list of publication, you can go to ensemble.org/info. In the About section, you can find the publications on the left-hand side. And with this, I'd like to thank all of you for your questions, and definitely the entire Ensembl team that makes this work possible, and also our funding bodies.
Okay, thank you very much, Alina. So we have had many questions during the webinar, which George and Louise have been busy typing answers to. I just wanted to highlight, we had a few questions about the recording of this webinar. Yes, we will absolutely send you all a link tomorrow with the recording for today. So you can rewatch or you can share it with colleagues who you think may be interested. So I think if you're able to just jump onto the next slide while everyone's thinking of any more questions they have. I just wanted to highlight that we have a couple of webinars coming up soon. So we actually have 2 Uniprot webinars, the first of which is happening tomorrow. But do go on the training website and check out what we have coming up. And then we also have a webinar next week on PDBE. So it's a protein data bank in Europe. Okay, so with that, I think we can jump to the questions. I think a lot of the questions have already been answered, so that's great. As I say, George and Louise have been very busy, but I just wanted to check in with the 2 of them and see if there's any questions that they typed an answer to that they think may be of interest to the whole group and to discuss out loud, if there's anything that stands out to the 2 of you.
Sure. So there is a question in the chat about are there any reductions for students in weekly courses? And I'd let you take that one first, and then I can talk about Ensembl.
Yeah, so we have many courses that EMBL-EBI training run. There aren't specific discounts for students. However, all our courses now do have bursaries available for a small number of participants in each course. So if you check out the EMBL-EBI training website, if you find a course that you're interested in, each course will have details of the bursaries available and how you can apply for them.
And in regards to Ensembl, you can definitely get in touch with us on helpdesk@ensembl.org. This is available in the slides that Anna shared with everyone. So Ensembl courses are free. So if you host us at your institute or if you join any of our open courses that we might do in the future, these are online virtual courses, these are free to attend. So we don't charge any fees. And there's a question on how to use it for plants. Okay, so let's go back to Ensembl's homepage and I'll show you the different species that we have available. So you can go to species selector. One thing that I forgot to mention is that you can manage the species that you've selected. So it's going to remember that in that previously I selected human and mouse, so it's going to remember that. So you need to manually go and remove these species if you're no longer interested in them by clicking on manage. And then from here, you can just remove from list by clicking on this bin icon to delete them. So let's go back to species selector again by clicking on this app icon at the top. And for plants, we've got various different plants species available. As I mentioned, we have rice over here. So we've got 27 different cultivars and then we've also got red wheat. Underneath here, we've got tomato. And then we've got, sorry, my Zoom navigation bar is blocking it. Then just over here, we've got soybean, and then we've also got malaria. So we've got malaria parasite, so Plasmodium. So you can select whichever one that you're interested in. Just for metazoa people, we've also got fruit fly. So we've got Drosophila here. And then, as I mentioned, that with we are constantly adding new species, and we've already brought in lots of species from rapid release to this site, and they include plants, they include metazoa and protists, and all of the different ensemble components. So for plants, let's click on red wheat. You've got different wheat cultivars over here. So we've got information from um, we've got the IWGSC, but then we've also got the 10 wheat genomes, 10K wheat genomes here as well. So you can select the ones that you're interested in. So for example, if I select this first one, and then if I click on add, it's become available at the top. And then to find out more information about it, you can click on red wheat, and then it's going to give you further information on the coding genes, the non-coding genes, and so on. You can also then view it in the genome browser. So you can search for a gene and then view that gene in the context of the wheat genome. Or you can also view it as the entity viewer. So you can look for a transcript or a gene ID or a variant that you're interested in, and then find out more information about it and download different sequences or different types of data related to this wheat gene or transcript.
Great. I think we're running out of questions because they've been answered so efficiently by George and Louise. I also just wanted to raise one that you answered already about the time frame for the new browser, but within that question, someone was asking as well about revising their teaching materials. And I just wanted to mention that there are lots of teaching and training materials available via both the EMBL-EBI training site and also the Ensembl, the current Ensembl site, that may well be of use for you when you're developing materials to use with your students. And those materials are made available under a license, which says you're very welcome to use them. You just have to mention where you got them from. So hopefully that will help you when it comes to revising your materials. And I'm sure there'll be lots of new Ensembl materials coming along soon to help assist with that in the future too.
Just one last question if you have time.
Yeah, absolutely.
So someone on the chat has asked about how to get gene ID. So there's different ways. You might be working with a database. So for example, if you're working with a gene and you are on the UniProt page for it, then within UniProt you might get the Ensembl matching ID for it. So that's one of the ways, and then you can search for that ID in Ensembl. Another way would be that you can also simply just look for, if you know the gene name, you can then simply just search for that gene name in Ensembl, and then within the results, we're going to give you the matching ID for that gene name. So that's how you can also get the gene ID.